Hi, welcome to this advertising video about our exciting PhD project. I'm Dr. Olga Kopach, a lecturer at the Department of Molecular and Biomedical Sciences. My research is focused around studying synaptic plasticity in the brain and understanding fundamental mechanisms of learning and memory, specifically focusing on interaction between neuronal and glial cells. I'm also interested in mechanisms of nerve cell dysfunction in ischemia, dementia, and other neurodegenerative disorders. For my research, I implement a range of neurophysiological techniques, specifically electrophysiology and fluorescent imaging techniques at different levels. In vitro, at the level of individual cells, in situ, within the organized tissue, and in vivo, within the intact whole brain.
I'm interested in molecular and cellular mechanisms that underlie cell signaling, neurotransmission, memory formation. To be able to visualize molecular and cellular processes that offers a very powerful tool for their investigation. And in order to aid this, I developed fluorescent biosensors for calcium, glutamate, glutamine, whatever I can think of. And recently I was involved in developing a biosensor for the mechanosensing calcium channel piezo1, which is called GenEpi, which enables visualization of mechanosensation. This PhD project is about mechanosensitivity. This is a fundamental and evolutionarily conserved biological process, crucial for how cells detect and respond to mechanical stimuli from their environment. Surprisingly, investigation of mechanosensing is a young research area. piezo1 and 2 mechanosensing calcium channels were only just discovered about 15 years ago. With the advent of GenEpi, it is now possible to approach investigation of piezo1 function by optical microscopic techniques. As piezo1 and 2 mechanosensors are calcium channels, GenEpi senses calcium by it being fused to a calcium sensor called GCAM. And therefore, when the piezo1 channel opens and calcium flows into the cell, then the fluorescence intensity of this GCAM sensor increases, and that's what we can detect by imaging the activity. The sensor fused to the channel, the construct is genetically encoded, which means that we can express and study it not only in cell cultures, but also tissue slices and even in living animals. For example, zebrafish. So the way GenEpi was developed is that we tested and selected variants of GCAMs, which means that their sequence was modified in strategically selected places to see how their biophysical properties are affected by these mutations, and I measured these biophysical properties. So we can measure the binding affinity for calcium, for example, in the fluorimeter, in which we shine light onto the sample and then measure the light that is emitted. And if we do this in a so-called stop flow machine, where we can mix together two small aliquots in syringes from two different solutions, we can measure how fast the calcium binds and how fast it dissociates. And you can see in the data that by making single point mutations, we could have quite major consequences in terms of changes to the kinetics. And so we aim to develop sensors that match the phenomenon that we want to study. For example, nerve transduction is really rapid, really fast. Therefore, we need fast neurotransmitter sensors, among other things, that would enable studying neurotransmission. And so for the mechanosensors, GenEpi is really the kind of first generation sensor, so it needs further development. But moreover, there isn't one in existence for piezo2, which is actually the subject of this PhD project. So therefore, an important part of the project will be to develop and characterize GenEpi2, let's say, for the piezo2 channel. So we will offer rigorous training in research methods. In this project, the range of techniques includes molecular biology to develop research tools, fluorescence techniques to measure their properties, and high-end neurophysiology, in which Olga has expertise and is her speciality. I am an experienced supervisor. I have supervised a number of PhD students, master students, dozens of undergraduate final year project students. I always have a little advantage over the students that I know what the final product should look like, and therefore I can guide you towards producing a high quality dissertation, make sure it's submitted on time, and get a nice publication out of it too. What do we expect in return for all the training and guidance? We just expect a strong interest in the research topic, a willingness to learn and understand the techniques, data processing and analyzing, appropriate note keeping, very important. To get through all this successfully, you have to have a strong motivation and determination. And as this is a lab-based project, on-site working is essential. There is a lively research student culture at the Tooting campus of our recently merged City St. George's University. This is where the old St. George's Medical School was, and this is where we run the health sciences courses and carry out biomedical research. So we have a departmental research seminar series that are with external and internal speakers, and so that we widen our knowledge and understanding of knowing what's happening, what are the most recent developments in research. Then there is an annual postgraduate symposium here, actually held in central London, usually very, very nice, for the postgraduate students to present the progress of their work. And also we have a research day, in which not just students, but all levels of researchers present their works and where prizes can be won.